But despite all that, I think that the field is still young, and the biggest challenge is still ahead.
The current sterility tests which are in place are just taking far too long.
Processes become shorter and shorter, and I think that there are more than one solutions to this equation.
Good morning and welcome to Miltenyi Biotec's All Things CGT vodcast. I'm one of the co-hosts here, James Havseth. I work in the communications department of Miltenyi Biotec. I was a long time ago a chemist, but that is why I'm joined by Silvio Weber, who is my co-host here as the more biological expert. And Silvio, I'm sure you can introduce yourself without my help.
Sure. Silvio Weber, biochemist by training, worked in immunology for a couple of years, and I'm now dealing with immunotherapy since 8 years at Miltenyi Biotec.
Okay. And today we are joined, thank you very much, by Grigory Efimov, the Scientific Director of Immuno and T-cell Therapy here at Miltenyi Biotec. You lead a multidisciplinary R&D team and are our manufacturing expert for today, but your team develops integrated tools for cellular immunotherapy manufacturing. Welcome.
Thank you.
And we're also joined by Marsilius Mues. He is the Scientific Director for CGT Analytics, our analytics expert today. And he oversees here at Miltenyi Biotec the establishment of multiparametric immune assays for functional characterization of engineered T cells, amongst other things. Welcome. Okay, shall we dive right in? We've got a few questions for you. We start with the topic of automation within CGT. So let me start with you, Grigory, here on this side. What role do you think automation plays in the context of CGT manufacturing, and how far can we push it?
Yeah, right. Let's start with maybe the most obvious, the simplest case. Of course, the automation is the way to reduce the manufacturer's dependency on the manual labor, highly specialized labor, which is also it's an expensive one, and people have to be substantially trained, and they're not easily available everywhere. So it's also location dependent. Mm-hmm. And of course, by doing that, you reduce your by bringing it instead of human to the machine, you reduce your cost of goods. But this was the simplest case, and in my opinion, not the most important one. What is more important, I think, is that the automation is the way to reduce the variability of the process. And in cell therapy, in particular autologous cell therapy, the variability is intrinsic. We're all humans, we're all different. Our cells are also different. This is true for healthy donors, even more so for patients, with all the disease history, all the treatment history, etc. So at the minimum, your automation is the way not to add up to this variability, not to exacerbate it. And for that, we have, of course, the industry have developed solutions. We as a company have solutions on the market that you can bring your fixed protocol, individual units, transfer it from human to the machines to reduce even further variability. Right, but you can also think of it as a way of mitigating the intrinsic variability of your starting material. And the way to do it is to introduce this closed loops where you have some kind of readout, analytical readout. I'm sure, Marsilius, you will talk more about it. And you couple it with the control of your process. So again, simple case is that you look at the numbers of your target cells in your culture, let's say number of your CAR T cells. And based on the readout, you decide how you formulate, how you fill your product. You do the same with the cell separation. You look at the number of your target cells and you decide how much volume you transfer to your culture. Okay. This is relatively simple. And again, there are solutions for that. Next step would be to implement adaptive manufacturing with this same closed loop approach, where you can look at the metabolic readouts, you can look at the cell phenotype, and you can think of actually modifying your culture, doing cell different feeds, media exchanges, all that, to be able to get to the product you need. Also, this is true with the flexible harvest. You look how your culture behaves over time, and when you have enough cells in your culture, you can stop and you can harvest the cells. So this is number 2, and I would say that there is additional aspect that with the automation you can also bring your manufacturing process closer to the patient in the more decentralized point of care way, which will have an influence on, of course, accessibility. It will reduce vein-to-vein time, so the time after the cells have been collected from the patient and the patient waits for the therapy. And it will also influence your costs additionally, because it will reduce the logistics and the expenses associated with that.
Okay.
I think there was a very broad response, and it resonates a lot with that, what I think. So maybe I can give you a segue here, Marsilius. So you were talking about deeper characterization of cells as one mean. I think that's totally where we have to go. But then the analytics might be very complex for this deeper characterization. I think for cell viability, we already have some well-automated systems. So Marsilius, what do you have in mind there if it comes to more definite, more comprehensive characterization? How can we tackle that?
Yeah, so I, of course, can also follow suit here with Grigory that cost is a very big problem here, but also for the assays, which are usually still for living assets as we have them here, are still very complicated. We need to make things as simple as possible. And I also fully agree that it's very important to characterize the products you have or the manufacturing you're doing in as much detail as is possible. I would, however, go for the way to distinguish that in kind of two areas where you say that for the simple manufacturing, so only to look at the QC of the cell manufacturing, you should have your assays as simple and lean as possible. It's a different story when you, of course, the one thing is like you would say you would like to learn more about your drug product. Then, of course, you want to have comprehensive and very big assays where you can learn as much as possible. However, for the QC itself, I would try to keep it as simple as possible. It's also like it all connects to the validation effort you have of that and how much it affects the routine which you're using in your normal QC environment.
And that's a good point. However, I assume if you would have one platform that could serve both of these issues, that would be very nice, right? Because if you can use it for more research purposes, but also for routine purposes, it can maybe answer both of these questions.
Yeah, so that is, of course, very true.
Yeah.
Of course, you would like to, in all directions, reduce complexity. So if you have something that can be transferred from the one to the other or the other way around, of course, that makes your life way easier. And having platform approaches, of course, here is a very good way to go forward. Yeah.
Brings me maybe to the second question, and you already referred to that point regarding more decentralized manufacturing, right? So different settings, we are all expecting that this is going to happen in the future. It's more a question of when rather than if. And so automation, there's also a downside, yeah, let's be frank, which is investment. So in the beginning, you have to invest into that. How do you make sure that these systems which you are automating are future-proof? Because changes, once you have set up your system, might be quite costly. And worst case, you even have to start from scratch.
Yeah.
So what is your approach to that?
Yeah. Very good point. And I also would like to segue here because Grigory mentioned like the normal big manufacturing, but also the point of care manufacturing. And we probably need to look at that like from two different angles. So when you're talking about commercial big scale manufacturing with many thousand doses per year, you probably would like to go to a full automation where you might even be able to parallelize your analysis. And then you have like samples run in parallel in a big high throughput. That is something which I could imagine or envision for commercialized big scale manufacturing. Point of care, I think, is a slightly bit different. There, I'm not really sure whether you need to have like this massive big robots that are doing everything. But here, it's even more important that the assays are very lean and simple. On the other hand, of course, also here, automation will help you a lot here because, as Grigory already said, manpower is costly and it's also like sometimes tricky to get the actual people, even if you have the money. So here, also to find ways of automation and, as Grigory already mentioned, maybe find ways to integrate both, to integrate the analytics and the manufacturing, to have that somehow combined in a system. In best case, they even talk to each other. And then you can have like the analytics run side by side with the manufacturing, and in principle, it goes hand in hand, and you can reduce actually the loads on the operators.
Yeah.
Which would also help you with a chain of custody, chain identity.
Absolutely, yes. And it actually, I completely agree, and it resonates quite well with your initial question, how far we should push it. And this would be, to me, I think the right way is not the how smart, that we should be smart about it, and that we should ask the questions, that we should look at our product, that it's efficacious, it's safe, and we should look at the process, that we maximize the efficiency of the process, we maximize robustness and reproducibility of the process. But I think we shouldn't blindly just follow automation to the extreme, because the extreme cases would be, like you said, this robotics complete automation, which will be probably very site specific, and it would not be easily transferable and scalable across different locations. So you have to be, as I said, smart about it, and ask how you optimize your process to the extent that you still have some flexibility and future-proof capabilities.
Mm-hmm. Sounds like the scalability might be the key challenge with this. So let's move on and talk about the interplay between process and analytics development. Grigory, let's start with you again this time. What is the interplay, right, between process and analytics development? How does one really influence the other? And yeah, can you talk us through that?
Yeah, of course. So we already touched a little bit on the future-proof and evolution, and it's probably true for both. For the manufacturing process needs to evolve, and also, of course, the analytical process needs to evolve. But they shouldn't be two different evolution tracks. They should be it's a co-evolution, right? They're intertwined. So we have to design our manufacturing workflow with the analytics in mind, asking ourselves, when do we sample, what do we sample, and why do we sample that, right? And same is true vice versa. When we're designing analytical workflows, we should keep in mind what impact it has, this particular readout, what impact it has on the process, what kind of control we're implementing there. And it starts with very early in the process development, with identifying unit operations, critical unit operations, looking at the requirements for specific unit operations. So what is the input, how many cells, what phenotype, what are the ranges, what is the output, again, number of cells, concentration, viability, right? And you're looking also at your rate limiting steps, and you're looking at your sources of variability to understand what you want to measure and how this will influence your process. Because you're not just measuring it, as Marsilius said, not just for the sake of it, but to really get hold on your process to have a better control on it. Do you want to?
Yeah. No, very, very, very well said. So of course, and when we're talking to customers or on conferences or wherever, you can see that still there is a lot of things measured, maybe not even for the sake of it, but there is still room for improvement, and you should really only measure the things that are really necessary for your process. Mm-hmm. Now, however, and saying what Grigory already touched on before, if we in the future might come to adaptable processes, where you said media change or the feeds or whatever, but also like where you can see, we could imagine that you are like giving additional goodies to the cells for rejuvenation, whatever you can imagine, that in principle, in the end, you get a better product out of it. And that also individually adjusted to the cells. That, of course, then in turn would need a more complex manufacturing, but also more complex analytics, which probably would then have to happen, at line all the time, not all the time, but really frequently, and then be directly fed back into the manufacturing system. So that is also something, if we are going that direction, then of course the analytics becomes more complex, but of course with a purpose again. So I totally agree that everyone should be like very considerate what they are actually measuring. And maybe one thing here also, which we touched upon in the beginning, is what you are measuring and to have the line of sight for later upscale, because we talked about scalability, so also for automation. And if possible, I only can recommend that already at the very beginning of any analytical development, it's already considered where the road should be in the end, where it should end. So if automation is the goal, which it most probably should be, because that's the only way to bring the costs down, that usually already needs to be designed into the processes or into the analytical processes at the very beginning, to be able to then, at a later time point, if not already implemented very early on, at a later point at least, then transfer those assays to automation. If that is not considered, it usually becomes very tricky then to have assays that were not optimized to build for it adjust later.
Yeah. I mean, it's a very compelling scenario, and it reminds me a little bit of the last conversation we had with our regulatory expert, Bernd Schröder, where we were thinking about innovative avenues towards regulation. And I think you would also need this if you follow this scenario. And there we were also a little bit trying to understand what are the red lines, let's say the limits in terms of simplification, risks, and so forth from a regulatory perspective. So how do you see that more from a technical perspective? Yeah, these scenario, risk, safety for the patient. Yeah, what do you think is absolutely crucial in terms of analytics, and where do you think we can really take innovative approaches, Marsilius?
Yeah. That's obviously a tricky one. But for example, like safety will be a given. So that will be, I would say, non-negotiable, like sterility testing. But however, here also, not only talking about automation, I don't have to tell you that, the current sterility tests which are in place are just taking far too long. And especially when manufacturing processes are supposed to become shorter and shorter, this needs to be adapted as well. So if you say, where's the red line? I'm sure that safety tests like sterility testing probably will need to be in place. But then on the technical side, we need to move things to make them more adapted to where we actually want to be in the end. Things like vector copy number, maybe that's something that might be negotiable one day, where we say we have now really enough process understanding. Maybe you can jump on that later as well. But if there is enough process understanding here that we really know what is happening in my manufacturing device, that this becomes a controllable asset in the end, that would be great. And I mean, the big one, it's always the potency testing, which is always a tricky one because it's one of the most complex assays. It's a bioassay. So also here, I see different roads. So the one thing also here would be more automation, but also like simplification of the assays. Somehow having more control of, for example, the target cells. If you're using one in a bioassay, that would be great. And of course, it would be even better if one day, because at the moment we still have this big issue in the field that the potency assays, they are not giving any clinical correlation, that here we might need to reconsider whether these functional, very complex bioassays are really giving us the answer we want to have, or whether we find here similar but way easier phenotyping assays for our QC testing.
Which could ultimately mean that regulatory, let's say, the idea behind could become a little bit more scientific, if I may say that. Yeah. Is that the idea behind?
Yeah. Would be highly appreciated. Yeah.
Grigory, anything to add?
I want to also stress that for cell therapy, in particular, autologous cell therapy, additional safety concern is the traceability or chain of custody, that you absolutely make sure that you follow the cells from the moment they're collected from the patient till the moment they're injected back. That there is no mix-up, because that's non-negotiable, as Marsilius said. Right, and this, of course, is also done with digital connectivity and with the help of automation as well. Then on the vector copy number, of course, I agree with you. When we know process more, maybe we can eliminate this testing. But I mean, the one way is to test it, of course, but ideally you want to avoid that you have the batch failure because you missed your requirements. You're not in spec. Right, so and the measures that we have to control that is the vector quality, potency, and also understanding of the process and influencing the process, the time that cells are in contact with the vector, the state they are in. So there should be also the improvements in the manufacturing process in order to control that, because of course this vector copy number is a genetic risk, genetic burden that we need to control. And actually the same with sterility, we need to measure, but we also need to active measures to control it. And the way to do it is the closed system, completely closed process, and aseptic connectivity of the reagents and components. And maybe one additional thing to consider from the safety perspective related to the manufacturing process is that there are the critical quality attributes of the product, such as composition, impurities, and also it can be the ratio of certain cell populations, which we also need to take into account in terms of safety.
So you mentioned some improvements that are necessary in the manufacturing space, no doubt also in the analytics space. Might be a good time to indulge in a little bit of crystal ball gazing. If I can ask you how you think that the field is going to develop commercially within the next sort of 5 years, and let's start closer to home and see if you see any changes that are urgently needed or urgently predicted within the current landscape, and then we'll move on to asking for the big predictions.
Yeah, maybe I can start here. So I think that, again, starting with the simplest, there are still some manufacturing processes, which I would say they're legacy processes. They are still, to a large extent, manual. Industry understands that it has to go. It has to change. They have to be transferred to the automated systems with reproducible outcome. And also, it will change the way that the costs are allocated. In particular, the influence of the human labor will decrease over time. So this is one direction, but this is fairly obvious. Another one that we also discussed a little bit is the shift from the centralized to the decentralized manufacturing. It can be regional. It can be also even point of care, bringing it as close to the patient as possible. And this needs to be solved in order that the patients in all the geographies get access to the treatments that they need. Because the current model of highly centralized manufacturing will not work in these markets which are underserved, which are not North America and not Western Europe. We need to have different models there. And point of care is one of the ways that we can reach the patients in this geographies. That would be number two. And maybe the last point that I want to make is that we see the trend that processes become shorter and shorter, and I think it will continue. Looking in my crystal ball, right? Even going to the extreme, to the, let's say, 0-day process complete in Vivo. And I think that these approaches will definitely find a niche in the therapeutic space.
Marsilius?
Yeah. So I also would like to start out simple, even though it is not simple. It is lots still of the analytics which are done for the QC testing for the CAR T cell space now here. Still, many of these assays are still way too complicated. They are done out there. And just like one simple example for flow cytometry, there's still far too much manual pipetting. So these things need to be like streamlined. You need like ready to go cocktails, which make your life just way easier. So everything becomes faster, cheaper, but also less prone to any human errors. I would like to touch again what we said, because there's two ways. So the one thing is, as you said, the processes are going shorter and shorter. That's one thing, of course, where we also, as I already said before, need to streamline the analytics processes. However, if you also go in the other direction, where we say that we might want to have actually the manufacturing to make the cells better, that, of course, will need more elaborate assays, but then probably also, and I see that then as a big asset, is certainly coming artificial intelligence coming in to help us, because it will become so complex, to help us understand the process, the analytics, the whole interplay, how we actually can get a better product out in the end. And probably also, when we are talking about decentralized manufacturing, that we get a much better control, or not, maybe control is the wrong word, but a much better oversight, what is happening in all these different locations. So probably also here, which goes towards a direction of AI again, but in principle, talking about software tools. So that is, as you already said before, in many places, analytics still is all happening on paper. That is something that certainly needs to go. And I really can imagine that we have like big overarching softwares that are taking control of the manufacturing process of the analytics and bringing those all together. So that is certainly things where these are heading. Maybe one last thing here, as you said, manufacturing processes are getting shorter and shorter. That is certainly true for the CAR T cell field, but of course it's much bigger. So now we have our mind focused on CAR T cells, but more will follow here. So I also see many other big manufacturing processes coming in, then for other applications, which then, of course, in the end, they will need similar but yet still different analytics as well, which will come anew, but then also need further adoption and changes, and later, also automation.
Yeah, right. I mean, many different patients in need, right, beyond the classical CAR-T indications. Shall we already sum up a little bit what we have talked about, or do you have some additional questions?
Well, if we've got a minute left, actually, I would like to see if I can push the both of you slightly outside of your comfort zone on this one, because we talked about in the next 5 years, and I know you have to be kind of safe about that, but I was once told that if you're looking at anything further than 10 years out, no one's going to hold it to you. However, I also know for a fact that several decades ago, the then CEO of IBM was on record as saying he saw a market for maybe 4 or 5 computers globally. So in that spirit, that it's completely acceptable to be vastly wrong about it, are there any predictions, for this space moving forwards 10 years or into the future? Or perhaps it's just where you would like to see it going? Either of you.
Maybe I can start. So I, of course, also see many things moving to in vivo. Many things moving to in vivo, and also then later new manufacturing processes, which still might happen in the next 5 to 10 years outside of the body. Mm-hmm. I also could imagine that those are moving more and more towards in vivo applications. And then, of course, the analytics they need to follow suit. So we will have something that will be given to the patient. So this needs to be much more characterized before it's given to the patient, the virus or whatever it will be then in the end. But also then we certainly need to very closely follow the patients after they received the whatever it will look like drug. Mm-hmm.
Right. Maybe to add to that, I think that despite all the successes that we saw in clinical trials, and also we have now a number of approved products, and we've seen that these products, these cells are tremendously efficacious, right? They are giving the treatment options to patients who previously didn't have any treatment options. And in some diseases like in autoimmune space, what we see is, we can call it even functional cure. But despite all that, I think that the field is still young, and the biggest challenge is still ahead. And this challenge is to see that, also to make sure that these therapies really reach the patients. This is a question of medical access, but you cannot solve this part of the equation without the second part of the equation of making it commercially viable. That the pharmaceutical companies have to understand how to make profit of this type of service. So I think the biggest challenge for us as an industry, as a field, in the next 5 and even 10 years is to solve this, to make it to solve this equation, to make it both accessible and commercially viable. And I think that there are more than one solutions to this equation, and in vivo would, in my opinion, definitely will be part of it. But I still think that there is a large space for ex vivo manufacturing, with maybe even emerging cell types other than conventional CAR-T cells, and that different teams, different companies will have different solutions to this equation. But this needs to be solved.
Okay. Yeah. Then that perhaps might be a good point to wrap up.
I think was already close my summary, right? No, but I think, really, I think automation is a key lever, right, to bring these medicines to the patients, like you said, and it needs automation on both ends, obviously, manufacturing and analytics. And one thing I think that really stick with me, Grigory, what you said, it's of course bringing down the labor costs and the complexity, but it's also about making sure that you decrease the variability. And this comes on many different layers, right? We also have raw materials which we bring into the process, which are biologics. So they also have certain variabilities going together with this autologous starting material. And so this can create effects that are very difficult to predict and also to control. And therefore, the next step, which is then more adaptive manufacturing, trying to enter the next level of control without automation, will not work. So this is very sure. And so then the next thing that is really key for me, that we don't develop that in silos. And that's easily said, right? Because, you're an expert in analytics, you're an expert in manufacturing, so you have your kingdoms on your own. So bringing this now together, infuse that, really organically, I think it's a challenge. Not because you don't want to work together, just because you have so much on your plate. And that is something, if you have an ecosystem where you can bring things together, this is very helpful, because then you can think things from the end already in the beginning. And this is maybe another take home, thinking from the end. Yes, crystal ball, 20 years is too far out, but I think the next 3 to 5, maybe even 10 years, we can predict. So we have to take into account that our automation systems, they can still evolve. If we don't do that, the investment is simply too high, and then the commercial viability will not work for the companies who are sponsoring these medicines. And then if you want to look even further into the future, I think new interfaces with AI and robotics, they are absolutely crucial. I think we still need to understand that on many different levels, also regarding regulation and quality control, things like that. But we have to start it now, otherwise others will start it. So with that, I would like to say thank you to every one of you here. I think it was a really great conversation. James, some concluding
Indeed, I mean, it seems like there is a lot of work for the both of you to do, Marsilius and Grigory, so we should probably let you get back to it. But yes, let me echo that. Thank you very much for giving us your time today.
Thanks for having us.